Hi, I'm Isabella Gesino, a junior pre-veterinary medicine major at the University of Delaware with minors in One Health, Equine Science, and Equine Therapeutics. This summer, I am working under the guidance of my mentor, Professor Aditya Dutta. His research focuses on reproductive health and early ovarian cancer detection in chicken models. This research greatly aligns with my personal ambitions and interests.
I'm Aditya Dutta. I'm an assistant professor in animal and food sciences with joint appointments in biological sciences and medical and molecular sciences. In the world of biology, nothing beats hands-on experiences, and so it's really important to take the time and the effort to train the next generation.
As a pre-vet student, I am fascinated with the concept of One Health, a holistic approach to public health that emphasizes the importance of the connection between human, animal, and environmental health. It was my father's battle with cancer in 2020 that inspired me to further explore this One Health concept. His resilience combined with modern medical treatment allowed him to defy his diagnosis. To me, this illustrates the growing importance of research that will pave the way to life-saving interventions, improving the health of all beings. As a pre-vet student, I am hopeful that one day I will too positively contribute to both animal and human health. My specific research project focuses on identifying the genes that play a role in the molecular fitness of ovarian follicles in chicken models using bioinformatics. Understanding why specific follicles are chosen in the ovulation process will aid in improving agricultural productivity and enhance IVF and fertility treatments in women. To better understand this research, it is important to know the basic biology at work. In chickens, we isolate samples of follicles within the ovaries. These are sacs that confer immature eggs to be ovulated. During each reproductive cycle, there are 4 stages of follicular maturity, known as primordial, primary, pre-recruitment, and pre-ovulatory follicles. At each stage, follicles are eliminated until there are only 4 to 5 remaining during the pre-recruitment stage. Finally, one follicle, the pre-ovulatory follicle, is chosen to ovulate for that cycle, and the egg is released to be fertilized. Our research compares the difference in gene expression by looking at the RNA between the pre-recruitment and pre-ovulatory follicles to see what makes that chosen follicle so special. We are looking for specific genes known as master regulators. We can think of these genes as the control centers within our genome. When these genes are turned on or off, they influence a network of other genes involved in a cascade of biological processes. Identifying these genes will allow us to make strides in improving fertility and agricultural production. In the lab, RNA is isolated from chicken follicular tissues and read using Illumina sequencing. RNA is read by incorporating fluorescently labeled bases, which light up as a laser passes through each cycle. This signal is captured by a camera and recorded on a computer's dataset. I analyze this data in RStudio, a coding software. This tool helps to analyze statistics and graph data into understandable visuals. With RStudio, we can identify pivotal genes that determine the biological fitness of selected follicles. To do this, we start with the quality control of the data, checking accuracy of our base reads, then align the reads to a reference genome. This allows the reads to be mapped to specific locations on a genome to identify the transcripts being expressed. From this stage, we are ready to start differential expression analysis, where we identify notable genes that are being expressed and that may differ between samples. Finally, we can make visualizations of our data to summarize the findings. Bioinformatics and programs such as RStudio allow us to work with large data sets and pave the way for groundbreaking discoveries. When we know what traits to select for in a genome, we can select for genetically superior chickens that contain the key master regulator genes responsible for follicular preference, and improve the production and sustainability in the agricultural industry to feed a growing world.